Let's embark on an incredible journey of interphase in the cell cycle. Interphase makes up the majority of the cell cycle and is divided into distinct subphases. In this video, we will explore each of its subphases in detail and understand their importance in the cell cycle. So, let's begin. As we know that the human cell cycle consists of two distinct phases, interphase and M phase. Interphase is the longer of the two, lasting significantly longer than the M phase as it is responsible for the preparation of the cell for division. During interphase, the cell grows and replicates its DNA in preparation for the division that occurs in the M phase. Now, let's have a look at both these phases in detail. Since these phases are quite complex, we divide them in sub-stages to understand them better. Let's begin with the interphase. First, here is an image that depicts the complete interphase in general. As we can see, the interphase has three stages: G1 phase, S phase, G2 phase. Let's begin each stage step by step. So, as the cell enters the first stage of the interphase, it's time for the first gap phase, abbreviated as G1 phase. This phase is the interval between the M phase of the previous cycle and the S phase of the current cycle, and it's the first stage of the preparatory phase before DNA synthesis. During this phase, the cell is still metabolically active and grows in size. All the organelles duplicate, and the building blocks that will be required in later stages are made. It's important to note that the G1 phase does not involve DNA replication. So, as the cell prepares for the following stages, it's growing, duplicating its organelles, and stockpiling materials for the future. Now, students, to make your preparation easy and complete, you should definitely check out the notes of all the three stages of interphase. And the good news is, EduRef provides notes of all the chapter in detail. During the second stage of interphase, known as the synthesis phase, or S phase, and as the name suggests, there will be synthesis of something, the nucleus and its genetic material, that is DNA, is synthesized, or we can say duplicated, in this phase. But that's not all. There is one more important organelle that gets duplicated in the S phase, which is centrosome, which is a bundle of threads present in the cell, which also gets duplicated. The centrosome contains smaller organelles called centrioles, made up of microtubules, which help in releasing the threads required for separating sister chromatids later. We will have a detailed look at this process in our upcoming videos. For now, all we need to know is that these threads are important for the cell division, and for this reason duplication of the bundle containing these threads is also important. All this happens in the S phase, so the S phase takes up the responsibility of duplicating the contents inside the nucleus and the centrosome present in the cytoplasm. Now, what's next? The next step in the process of the interphase is the second gap phase, abbreviated as G2 phase. This phase allows the cell to grow more. Along with the enlargement in the cell size, the cell also makes more proteins and organelles. The most important function of this phase is that it prepares the cell for mitosis. This phase helps the cell to prepare for the actual cell division in the M phase. So, now are we set to study the next phase, that is the M phase? No, not yet, because we should know one more intermediate step which plays an important role, whether the cell is going to have the M phase or not. Now, are you curious to know about that intermediate phase? This is the G0 phase, an intermediate phase between the M phase, that is cell division, and the G1 phase, that is growth. Also, there are two types of G0 phase: the quiescent phase and the senescent phase. The quiescent phase is when the cell temporarily stops dividing, but can return to the G1 phase if the external or internal factors that caused it to enter the G0 phase are removed. On the other hand, the senescent phase is when the cell permanently stops dividing. The G0 phase is a metabolically active phase where the cell is highly active, but it does not divide further. This phase is determined by a variety of intrinsic and extrinsic factors, such as resource availability or nutritional deprivation, etc. Let's discuss an example to make you clear about the G0 phase. So, nerve cells are the example which enter G0 phase permanently. They remain metabolically active but do not divide further in their entire life cycle as they lack centrioles. So, now you know what happens when a cell stops dividing. Fascinating, isn't it? In our next video, we will explore the second phase of this fascinating process of cell cycle, the M phase. We will go over all the details and explain everything you need to know in an easy-to-understand way. So, get ready to explore the M phase and the mysteries it holds. We hope that you have understood the topic. And now, you should start studying this chapter with the EduRev app as you get video lectures, notes, variety of questions, and MCQ tests for all the chapters of each subject. It's like having the entire school in your pocket.